Okay, everybody, we will get going for today. Thank you so much for joining us. I would like to begin by acknowledging the traditional owners and their custodianship of the land from which I join you today. On behalf of the University of Queensland, I pay our respects to the Yugara and Turrbal peoples, their ancestors and their descendants, and their enduring cultural and spiritual connections to country. I would like to extend that acknowledgement to the traditional owners of the lands from which each of you are joining us today. We recognize their valuable contributions to Australian and global society. Welcome, everybody, to the first lecture in the 2026 People, Plants and the Law lecture series. My name is Shruti, and it is my pleasure to be chairing today's seminar and chairing the series for most of this year. One of the aims of this series is to showcase some of the best work currently happening in the area of people, plants, and the law. And 2026 marks the 5th year of doing just that in the People, Plants, and the Law online lecture series. You can find all of the recordings from previous year's lectures on our series websites, and that is also where the recordings from this year's lecture will be uploaded. So in today's lecture, we're again showcasing some of the most exciting and interesting work being undertaken in this space from not one, but 3 important and influential scholars here. Today we have Claudia Calderón, Jean-Michel, and Jorge Contreras with us. Dr. Calderón is an affiliated professor at Universidad de San Carlos de Guatemala and a teaching faculty member at the University of Wisconsin-Madison. Her work advances just agroecological transitions through participatory and transdisciplinary approaches, centering ancestral knowledge, respectful collaborations, and mutual learnings between Indigenous communities and academic institutions. Professor Jean-Michel is a Villas Distinguished Achievement Professor in Bacteriology and Plant and Agroecosystem Sciences at the University of Wisconsin-Madison. His research explores the molecular mechanisms behind plant microbe symbiosis with the goal of improving agricultural productivity and sustainability for the production of food, feed, and biofuels. And finally, we also have Professor Jorge Contreras, who is a distinguished university professor and the James T. Jensen Endowed Professor for Transactional Law at the University of Utah's S.J. Quinney College of Law. His work focuses on intellectual property and innovation law, and he has written extensively on patent pledges and other forms of open innovation in the life sciences. Our lecture today will go for roughly 30 to 40 minutes, and we'll have plenty of time for questions and discussion after. If you have any questions for our speakers throughout the lecture, please post them in the chat and we will come to them after the talk. I will now hand over to Claudia to take us away.
Thank you. And I'm just going to share. All right. Are we good? Can we hear me well and see those slides? Perfect. So thank you so much, Shruti, for the nice introduction, and good morning to people in that side of the world. Good afternoon for other people here. Thank you for all the organizers for the kind invitation for us to share this space with you all. We are very grateful for the opportunity to be here and have this conversation. And I want just to start by saying that there's another member of our team, Jack Kloppenberg, who's now in the audience, but he's an instrumental part of our team. And I just want to recognize that up front. So the story we're going to tell today starts with a very special plant that it's muk. Mok, Inmije, Shob, Nuni, Inu, Ishim, Madamin, Metomen, Wewashesh. These are just some indigenous names of a plant that you all know as maize from Mexico, Guatemala, and Wisconsin. In the village of Totontepec in Oaxaca, a remarkable 5 to 6 meter tall plant that you can see here and that you're going to hear a little bit more later by Jean-Michel Ané's presentation has the capacity to self-fertilize by fixing its own nitrogen through bacterial associations. This plant is referred by people in Totontepec as Tzapop Mokshush. Later in this session, we will learn about plant microbe symbiosis, and then George will examine the legal approaches to seed germplasm transfers and the alternatives to the restrictions of intellectual property regimes. But first, let's focus for now in Totontepec and many other Oaxacan villages where for millennia, indigenous people have nurtured this plant. They have eat it, planted it, saving seeds, cooking with it, honoring in ceremony, and selecting the traits that have made this plant resilient and capable of fixing nitrogen. In the 1980s, a renowned microbiologist from Mexico arrived in Totontepec, and he was the first to characterize the mucilage and the nitrogen-fixing bacteria that it hosts. But because of the uniqueness of these traits and the plant and its potential for agriculture, scientists from the United States, particularly from UC Davis and the private company MaSE, began visiting Totontepec. Later, researchers from the University of Wisconsin were invited to join the effort to study capacity of nitrogen fixation from this plant. However, the way the research was conducted, the processes for collecting plant material, and the agreements that were made between MaSE and the local authorities raised very serious concerns. And the situation set off alarms across Mexico and beyond as a potential case of biopiracy, highlighting urgent questions about ethics, sovereignty, and the protection of indigenous knowledge and biodiversity. So over the past 6 years, we have been working with Jack Kloppenberg and Jean-Michel Ané through incredible collaborations with partners and friends, especially from Espacio Estatal en Defensa del Maíz Nativo de Oaxaca, as well as academics and people from rural communities in Oaxaca, to weave networks of exchange that recenter ancestral knowledge and wisdom, and to invite communities, academic communities, in respectful and reciprocal learning. It's not always easy, it hasn't been, and it doesn't always work. Our project is only scratching the surface of these conversations and exploring ways to honor these different ways of knowing and operating. So our project that we referred as MaSE, Maize Sovereignty for Everyone, was trying to open conversations about research involving native maize and to shift the narratives so that indigenous ways of knowing are central and not peripheral to agricultural education and research. So you have in this slide some of the core aims of our project. And basically, we were trying to bring together indigenous communities and academic partners from Wisconsin and Mexico via bilateral cultural exchanges to reflect on the science and ethics of research our own native seeds, and doing so by strengthening North and South indigenous dialogue around seed sovereignty. We were interested in building culturally grounded partnerships that foster horizontal, respectful exchanges of knowledge, and also co-develop culturally acceptable guidelines that bridge Western scientific paradigms with indigenous epistemologies to support the use and conservation of indigenous biodiversity. So in 2024, our group published this article that is titled The Nagoya Protocol and Nitrogen Fixing Maize, Close Encounters Between Indigenous Oaxacans and the Men of Mars. You have the QR codes for the English version and a Spanish translation of that article. And basically, in that article, we're analyzing the access and benefit sharing agreement with the Nagoya Protocol that was negotiated between the community of Totontepec and the private company MaSE that was interested in accessing those nitrogen-fixing maize. So our analysis what revealed basically that there's a lot of weaknesses in the framework of the Nagoya Protocol framework and the access of benefit sharing, and as well as the current governance systems for genetic resources that was in place in Mexico. And we also point out the limitations of the framework of the Nagoya Protocol and its ability to prevent biopiracy. So at the end, we are calling for more ethical and robust legal frameworks that truly protect native seeds and recognize the value of indigenous knowledge. So we talk about Nagoya Protocol, and maybe not everyone in this audience is aware. You have heard, but it can be a little bit obscure. So for decades, there has been always an imbalance between the wealthier countries, larger corporations that are interested in bioprospection, and then the countries from the global south, usually that are resource rich, but who may not have the same power of research to invest in research. And that biological diversity that is found in the global south has been accessed, extracted, studied, distributed, stored in places in germplasm banks in the global north with very little information coming or returning back to those countries and to the knowledge holders, and also with all the seed and biodiversity guardians who have been protecting these plants or seeds or biological resources for millennia, and without any power to decide what is going to happen with those genetic resources. So a way to redress that issue came in 1992 with the Convention of Biological Diversity that established a international framework to preserve biodiversity, to use it sustainably, and super important, the fair and equitable sharing of the benefits that arise from using these resources. And as part of this mandate of the Biological Diversity Convention, countries were developing a binding agreement where they needed to establish access and benefit sharing to that was done by adopting this Nagoya Protocol. So under the Nagoya Protocol, you have on the purple side, you have a user, and it can be a country, it could be an institution, it could be a private company who's interested in accessing biological material from a provider country in the square, the blue square on the left. And in order for the user to access that material, there needs to be an internationally recognized certificate of compliance. So that certificate of compliance happens only when there's two things that happen. The permit, like the arrow that is in blue there, it requires prior informed consent, that is spelled by PIC, and also a material agreement transfer, where that is outlining how the resources are going to be used and the benefits are going to be shared. So in that scenario, if you have an agreement between the user and the providers and you have this clear explanation and agreement, then that material can move to the users. And in theory, that seems like a very good mechanism to share, to protect, but also to share the benefits to the provider country. Unfortunately, the Nagoya Protocol, in reality, results in very ambiguous certificates, and they are not transparent, and they hide under confidentiality clauses. So that particular scenario with the nitrogen fixing maize that we have been studying, that's exactly the case. We couldn't find any information. Everything was behind confidentiality clauses, and that is a deep flaw of the Nagoya Protocol. If you are a user and you are certain that you are giving a fair and equitable sharing of the benefits, in theory, you should be providing that information for everyone to see. If the information is kept confidential, how can the system and how can everyone else be aware, or how can the system be ensuring that there's no biopiracy that is happening? And the reality is that for the case of the nitrogen fixing maize, there's multiple references that I'm posting here. There's books, there's articles, there's even a recent article from 2026 that appears on the top right side of the slide that is recalling the narrative that was shared by the companies, by Mars companies, and by UC Davis, saying that the agreement was going to be a 50-50 arrangement. And the reality is that it was a 99 to 1 percent arrangement. You can guess where the 99 and the 1% was going. So the problem is that then that narrative continues to be repeated, even though that the agreement is, because the agreement is not public, people just repeat what they found online, and the online information is that the private company was giving 50% of the benefits to the community. So in order to better understand the issues of appropriation of biological material, we have set like a set of bilateral cultural exchanges between Wisconsin and Mexico, and that have occurred throughout the past 3 years to be able to engage in these conversations, to listen from Indigenous partners in both territories, to learn about the challenges of seeds. And we have had the opportunity to bring seed protectors, knowledge holders from Mexico to Wisconsin and vice versa, to be able to exchange and recenter those stories around seeds, native seeds, the challenges and the success stories, as well as recentering Indigenous voices in the academic circles. We have had plenty of discussions and opportunities to talk about what are the issues with the diversity and how it's been shared, how it's been protected, who is benefiting from the research. And it has informed the way that we have proceeded in our project to try to understand ways where seeds, after they are collected from the Global South, can be ethically used and shared. So throughout the past years, we have been discussing in this ethics of sharing, and I'm going to pass now the microphone to Jean-Michel for him to talk about the plant, and that is the center of this talk.
Great. So I wanted to first talk about the biology to show you the value of the trait in the maize from Oaxaca, and also to show you the extent of the problem that we have now of what we do now, how can we move forward. So first, I wanted to introduce the process that we study in that maize, and which is the process we call that biological nitrogen fixation. It's the ability that some bacteria have to convert nitrogen from the air into ammonium that then plants can acquire. There are some plants, legumes in particular, can associate with some bacteria we call rhizobia. And in legumes, that process of biological nitrogen fixation is extremely efficient. Legumes get 60 to 90% of their nitrogen from the atmosphere through bacteria through that process. That's why legumes are so popular in all agroecosystem around the world, because they can grow without any nitrogen inputs. In contrast, when it comes to cereal crops, most of our cereal crops like maize, sorghum, rice associate very poorly with these bacteria. And so it has been a goal of my scientific community for decades, actually more than 100 years, to improve association between cereal crops like maize and nitrogen-fixing bacteria, with the goal of Claudia, can you go next? Our goal of better using biological nitrogen fixation in cereals is to reduce the amount of synthetic fertilizer that we are currently using to grow our cereal crops again, like maize. Synthetic fertilizers, on one hand, are, well, first, they have been invented at the beginning of the 20th century, and currently they feed about half of the world population. So we absolutely we are strongly dependent these days on these fertilizers in particular for cereal production. But the fact that we use so much synthetic fertilizer also comes with negative consequences, and I'm sure that you probably have heard about some of them. Consequences on the environment. When these fertilizers are not used by the plants in the field, they contaminate underground water, and that leads to eutrophication and what we call dead zones, like in the Gulf of Mexico. So water pollution is one issue. Some of these fertilizers also get degraded into greenhouse gases, and they also overall degrade soil quality. So for all these environmental reasons, especially in the US or developed countries, people would like to reduce the amount of fertilizer we use and replace that with biological nitrogen fixation. At the same time, these fertilizers are also expensive, and fertilizers are made out of natural gas, and the price of fertilizer basically depends on the price of natural gas. And so these huge fluctuations in the price of synthetic fertilizers, nitrogen fertilizers, impact in particular developing countries. And so in developing countries, access to fertilizers and their price in particular is often limiting. So being able to replace synthetic fertilizers by biological nitrogen fixation also has a huge potential to basically help smallholders in developing countries increase their yield, because for them, access to fertilizer is extremely limited. Next. So as Claudia said, I was invited by the group of UC Davis and MaSE to evaluate nitrogen fixation in the corn from Totontepec. And the situation in Totontepec was fairly unique, because they traditionally growers in Totontepec grow maize on the hills of the mountain in the soil that is extremely low in nitrogen content. And they grow maize year after year. It's what we call continuous crops, without any inputs of synthetic fertilizers. And so it was really intriguing, and they came to me to evaluate how much fixation was happening really in these maize varieties. Next, Claudia. Thanks. So the first technique that we used for the project is called 15N natural abundance. And already in 2010, we obtained data from Totontepec that the maize was potentially obtaining about half of its nitrogen from the atmosphere. And then Pablo Zamora went back to worked in Totontepec in 2011 and 2012. And we got more data that during the season, showing that during about 4 months during the growing season, these maize accessions were obtaining about half or more than half of their nitrogen from the atmosphere. To give you some context, when we perform the same kind of experiment on the maize that we grow, let's say in the US, maize gets less than 1% of its nitrogen from nitrogen fixation. And so seeing data that maize was getting more than half of its nitrogen from that process was really surprising and really outstanding. So it took us many years to confirm, but basically we used all the techniques that are available in my community to evaluate nitrogen fixation, and they all showed that that maize was obtaining a very significant fraction of its nitrogen from the air, much more than what we honestly were anticipating. Next. And so what we showed in that during these years is that the process of nitrogen fixation is again performed by bacteria, and that it happens in a gel, we call that a mucilage, produced by roots that develop on the stem of the plant. So these roots are never touch the ground. They never go inside of the ground. So we call them aerial roots. They stay in the air. And when it rains, they produce a lot of that gel mucilage that you can see on the slide. I don't know, Claudia, if you can play the movie on the bottom left. Not sure that it's going to play well, but yeah, you can see literally mucilage drips from these roots. And so what we showed is that mucilage is really the place where we have that intense nitrogen fixation happening. And it's clearly a plant trait. These maize plants are unique in their ability to provide that unique environment, that mucilage that you don't find, or you find it far lower amounts in the accessions of maize that we use in the US, for instance. Next. So these plants, when you look at them, are striking because they have all these roots on the stem. You have many of nodes that carry these roots. You can find sometimes up to 10, 12 nodes with these roots. And when it rains, these roots, many of them produce that mucilage. So clearly the number of nodes carrying these roots is an important factor for the trait. Can you go next? At the same time, that maize from Oaxaca has traits that are unusual and not very well adapted to a wide range of environments. One example is the size. These original landraces are about 5 meters tall, and you may be used to seeing maize in a maize field. It's about 2 meters, 2.5 meters. Well, these ones are twice the size of a tall maize. And so clearly the size is unusual and makes that kind of landrace difficult to use in other places. Also, these landraces grow over 9 months, which is a really long growing season. And so it really makes these accessions impossible to use in temperate regions. At the same time, what I show you on the right, on the right is that they do produce seeds like a normal maize. And as Claudia said, in Totontepec and in the region, families use that maize for their daily meals. Next, please. So something that we realized a few years later was that the maize was not that trait that, again, all these roots on the stem and high number of nodes with roots was not unique to Totontepec. And so initially for the first at least 5 or 6 years, we thought that it was unique to Totontepec. And so in terms of, we can talk about that later, but in terms of providing benefits back to the community, it would have been simple if the trait was only in Totontepec. Well, working with collaborators and the CIMMYT in particular, we realized that the trait is present in the highlands of Mexico. So here on that figure, you can see where the village of Totontepec is, and around we found the trait in many other villages around in the Sierra Juarez in the highlands of Oaxaca. But we also find the trait in maize, which is in the lowland of Oaxaca, and also in basically throughout the region, even in Guatemala. So the trait is not unique to one village, it's throughout the region, which clearly complicates the effort if we want to give back to the community. It's not one village, it's the whole region. Next, please. So I wanted to show you what we do now and what is going on and why we think it's important for us to think about how we'll be able to share the material that was gifted to us. So first, regarding what we are really selecting for. So I told you that these maize accessions from Totontepec have many nodes on the stem with these roots. That's clearly important to have nitrogen fixation happen during a long time during the season. But we also identified that the diameter of these roots and the number of roots per node are really important plant features that allow efficient nitrogen fixation. And we are currently using these plant traits as a proxy to evaluate nitrogen fixation in the field. Next, please. So what we are currently doing is that we started a program to do breeding and genetics to understand the genes involved in that process with Dr. Natalia De Leon here at the University of Wisconsin-Madison. Natalia is a fantastic colleague, a plant breeder and maize geneticist. And so what we did is that we started as a starting material for our project. We are using seeds from the CIMMYT that we obtained through an SMTA, and also seeds from the USDA grain, which is a USDA bank in a seed bank in the US, and that one comes with no MTA at all. And what we did is that we started crossing that material that has the trait to accession that we call XPVP, that are inbreds, that are expired variety Plant Variety Protection Act from the Plant Variety Protection Act. And we took XPVPs that are inbreds well adapted to the Midwest or the United States. Overall, they are well adapted to temperate regions. And what we did is simply by crossing the landraces we got from CIMMYT and green to these XPVPs, we were able to introduce the traits that I just described into accessions that are now well adapted to temperate regions. So meaning the US, but also many places around the world. What we showed is that, well, breeding works, is that we now have accessions that look normal from an agronomic standpoint for people in the US. They have a normal stature, size. They flower within a short period of time which is well adapted for temperate regions. And so now we have plants that really look like the maize that we're used to grow in the US, and also have that aerial root trait. What we showed is that when the plants have the trait, they can obtain 35 to 40% of their nitrogen from the atmosphere. Again, if you compare that to the kind of inbreds that breeders are using here in the US, a normal standard corn in the US gets less than 1% of its nitrogen from the air. So we went from 1% to already 35, 40%. So clearly that trait is extremely beneficial even in temperate regions. And in maize, very often we work with hybrids. And so we started making hybrids with and without the trait. And now we have 2 years of data showing that we can reduce the input in terms of fertilizers by 80 pounds per acre. So that's a standard amount of nitrogen fertilizer in Wisconsin would be 220 pounds per acre. We can grow these varieties under 140 pounds per acre with no problem. And the hybrids that have the trait outperform the hybrids without the trait by 50 to 90%, meaning you can reduce very significantly the synthetic fertilizer inputs and get a much, much better yield. So of course, that's interesting for places like here, the Midwest, in order to reduce synthetic fertilizer and reduce all these negative and environmental consequences of these fertilizers. But also probably even first, that trait can be useful right away for developing countries in places where people cannot afford fertilizer. Right away, having the trait already has a very significant impact on the yield. So it's important for us now to think about what do we do, and that's the question that I asked to George and Jack Lopenberg and Claudia Calderón. How can we move forward? I mean, how can we put that kind of trait into the hands of, let's say, for instance, smallholders in Africa or in South America that cannot afford fertilizer because that trait is valuable. At the same time, Claudia clearly explained all the issues that are associated with how we even got that material. It was gifted to us. So how do we do, what could we do to share it?
Great, great. Well, so thank you. So this is where I come in. I am a lawyer. I'm not a plant biologist or a scientist, but I have spent a lot of time thinking about open science and how, over the last 30 years, different systems for conducting scientific projects in an open way, sharing scientific results, have developed, and what are um, the legal frameworks that we use to implement those types of sharing arrangements. And these range from data and software and patents, which we won't talk about today, to what I call open source bio and biomaterials, which is really the most relevant to the topic today. So when Jack and Jean-Michel and Claudia asked me to participate in this very important project, this is sort of my starting point. So we can go to the next slide. There are definitely a number of coalitions and projects around the world that have, for a number of years, been thinking about the sharing of seed resources and germplasm, including the ones here. And the one with which I'm most familiar, if you go to the next slide, is the Open Source Seed Initiative, which, again, I think this project shares the greatest heritage with, which has operated for many years under a fairly broad pledge, which says you have the freedom to use these seeds in any way you choose, and in return, you pledge not to restrict others' use of those seeds. This is, again, fairly high level. This pledge accompanies seeds when they're provided to a group, but the desire in this project was to try to create something that would be more legally robust and potentially show the seriousness of the sharing commitment that this project had. So the next slide, when we go back, we look 40 years ago to the open source software world. These people really developed a sophisticated set of legal instruments that enabled them to share software code while retaining their copyright in it. And so this is what became open source software. And today it's quite a huge phenomenon, everything from the Android phone operating system to the Linux operating system to quite a few very important other software programs are open source. And so what does that mean? If we go to the next slide, we can see just a list of characteristics of open source code that have been around, again, since the late 1990s. This makes the code accessible to others, modification is allowed, what you produce from it can be sold, it doesn't have to be given away, no restrictions on redistribution, and then an element called sometimes copyleft, sometimes share alike, which means if you get this open source code software, what you create and what you distribute later must also be distributed on an open source basis. So if we go to the next slide, we can see that this, has been applied outside of the software area to biological projects, access to enabling technologies of all kinds, with a very broad grant of intellectual property and allowing broad, decentralized, distributed development. Again, very similar to what we see with software. And in the next slide, we see how this copyleft notion works legally. It says basically that if there is an open source code component that you incorporate into a larger software program, well, then the whole thing, the larger program and what you incorporated into it, all become open source. And this was viewed as a way to perpetuate the open nature of some of these programs and to prevent somebody from taking open shared code and bringing it inside of a wall, inside of a paywall, so that others had to pay for it. It made everything open that used the open code. So if we go to the next slide, this idea was taken up by Creative Commons, which if you click, you will see the share alike moniker, which is used to designate online content, images, videos, music that is to be shared online in this same way. If I share it with you, then whatever you make from it, you also must be willing to share with others. And this is the mode that we decided to use in the in this MaSE project. So in the next slide, there is an important difference, though, between software and online content and seed germplasm, in that in the seed context, we are explicitly not dealing with material that is covered by patent or other intellectual property. So unlike software and online content, which is covered by copyright, this set of obligations and restrictions is only introduced by contract. There's not an underlying IP right, and that's very intentional. We did not want to depend on patenting of these materials in order to share them. So this is a slightly weaker version, admittedly, of the share alike concept, but we feel that in this context it works okay. And so if we go to the next slide, we can see the contractual types of restrictions that we attach to the seed germplasm that is shared. The materials are provided subject to this material transfer agreement. Whoever receives them can modify them and produce output, which of course are progeny plants or further seeds, but those modifications and outputs have to be distributed under the same terms. They also have to be open. Now, that's not to say that the recipient can't sell the outputs, the seeds, but not in the way that implies a patent type reach through royalty on future generations, only sale of the actual seed commodities themselves. Further, there's a restriction on enforcing IPR that might be embodied in either the original material or the modifications that are made or the outputs. And then finally, for the researchers, any publications that are produced as a result of these materials must acknowledge the source of materials, most importantly, the original source, which are the Oaxacan farmers who originated the material. And so to the next slide. As you can see, this is still something of a work in progress. We're very close, we feel, to a final material transfer agreement, but we are open to community input. And Claudia, I believe, has a version that she's willing to share with anybody who would like to give us their feedback and comments on it. And so that's really my piece of this. The next slide, I think we go back to Claudia.
Thank you, Jean-Michel and George. And just to quickly summarize, in an ideal world, seeds would be shared and used freely, unencumbered. Nevertheless, our work is taking place within an economic system where intellectual property right and laws govern seed transfer. Even the work at the university, we operate in a system that protects intellectual property. And in order to protect innovation and derive benefits, those systems clash with the very systems of the indigenous communities that were working, the traditional ways of seed and knowledge sharing that they have used. So as you heard during this presentation, our group is currently exploring this share-alike material transfer agreement to preserve that idea of sharing reciprocity, to share the seeds in ways that uphold at reciprocity and gift economies. These are the ways that we've learned and continue learning from indigenous peoples around the world, how their systems are rooted in communal practices and solidarity networks. where these seeds are circulating freely as long as they're planted, cared, and consumed with respect. But we also recognize that there are vulnerabilities to this system with climate change, aging farming population, biodiversity loss, and challenges in transferring knowledge across generation. So what we are looking for ways to continue these, inspired by our collaborators in different indigenous territories of the globe. And we want to acknowledge all of their contributions, particularly our friends from Espacio Estatal, Jody, Felipe, community members that hosted us and explained their connection with the seeds, Instituto Paz en las Americas, and you can see the rest of collaborators from the universities, from our universities, from Wisconsin, funding that we obtain, and many people who have worked in the labs with us. And I just have an extra slide here. There's many books that are available for people to continue learning, and these are some that we wanted to put forth for people to learn from. So we open these for questions and hope that we can engage in a good conversation. Thank you.
Thank you so much, Claudia, Jean-Michel, and George. That was incredible to hear about. If everyone would like to join me in giving another round of applause, I really thoroughly enjoyed your lecture so much. If anybody has any questions, would you like to put them in the chat? Or you're also welcome to unmute if you would like to say your question yourself. Just while everybody is collecting their thoughts, I will abuse my power as chair to ask some of my pressing questions. Thank you so much again. This is really interesting for me, and I really enjoyed hearing about that. Probably have a couple of small questions. With the share alike material transfer agreement is really interesting and a really impressive model. With the drafting, or like, have you received feedback from the Indigenous communities around that, or have they asked for anything specifically that they wanted included or not included in that? Just a question of curiosity.
Yeah, no, thank you so much for the question. We've shared that in one of our conversations we had last November with Indigenous members, and we've shared also with our collaborators in Mexico. The reality is that there's not a blueprint of how to work. The blueprint has been modeled with the intellectual property, and because the seeds have been collected from gene banks, and the gene banks act in modes that they extract and appropriate the seeds without giving a lot of agency to the donor countries. A lot of times the communities have said, look, these seeds don't come really from us. They have already been taken since 1950, and they have been extracted in ways that are colonial. And we appreciate that you are trying to prevent them from being protected, because the patenting was really a contentious issue. So bringing these as an alternative path, they say it's a way forward. But they say that ideally in the future, the research happens where research is started and initiated from within the communities, and where they can have more of a say of what is going to be happening, and more agency on the material and the collaboration itself. So with the share alike, we don't have a lot of feedback. We had some feedback in the sense of, let's give some of the the stories of these seeds and their traditional names, or we're trying to incorporate that in the way we are framing our conversations. Jean-Michel and George and Jack, if you want to add something, please jump in.
I would say that I shared that idea. I mean, I presented this last November at the it's called SMB Plant Biology Congress of plant biologists in Mexico, basically to Mexican colleagues working in plant biology, and many of them were very excited, were a bit surprised, I think, of the approach, but the feedback was very positive. Yeah.
Thank you. Thank you for that. We have a question in the chat, which I will read out. Um, Marita has given a wonderful question about, she's curious more broadly about your own normative framework. When you design or advocate for share-alike MTAs, what do you consider necessary for benefit sharing to be meaningful? Beyond preventing enclosure, what elements need to be in place for sharing to be substantively fair?
Yeah, thank you so much, Maria, for the question. We have thought a lot about the issue of what is fair, and we discuss a lot more in the article. So maybe Phoebe, if you have that link, you could share some of our reflections about that. And a lot of times, that's a little bit of what Nagoya tried to do, try to put a framework that would promote that equitable sharing of benefits, but there's no real definition of what is to be expected in those, what is fair and what is equitable. A lot of times these agreements have focused on monetary benefits, but a lot of times the indigenous communities they don't assign property to biological material. For them, the seeds and maize is sacred, and you don't assign a dollar value to a relative. So how do you reach a fair agreement? I think that that is something that needs to come from that same suggestion that the indigenous communities shared with us and our collaborators. When we engage in research, we have to have these upfront, very honest and transparent relations. And what are we going to be doing with the material? And what are the ways that we can share the information? How can we train people from the communities? What are elements that they're interested in engaging, so that that is written in the agreements and that can be done in future research, ethical research. So I guess that to answer your question, that should be something that needs to be done and seated in honest and horizontal conversations between the researchers or between the users of the material and the communities in terms of our share alike. We are going to share the seeds, and we are open. We have gone, as Jean-Michel mentioned, in conversations, in seminars and conferences in Mexico to open our labs, our opportunities to discuss, to have people from Mexico help us guide the next steps for our research.
Yeah, I would just add to this, just coming from the perspective of someone who has spent many years thinking about and participating in data sharing projects in the sciences, especially in the life sciences, that this is a somewhat new question in the field. And there have been primarily two motivating factors in these data sharing arrangements. One is sort of promoting the social benefit of maximizing access to data and promoting research to the greatest extent possible, right? That sort of underlies much of the open science movement. And then the second motivating consideration, especially when we come to biological materials, is has adequate consent been obtained from donors of either human tissue samples or the owners of whatever the resource is, and so there's a significant amount of thought and ethical writing over, again, the last 30 or so years that's gone into those two questions. This question is kind of new. It does add sort of a third component to that traditional twin set of considerations, and I think it's important, and again, but is not a mature area of scholarly exploration yet. I don't think there's a consensus in the sort of the data sharing community about any of these questions. And so this project, I think, is quite exciting from that standpoint, in that it will enable us to explore a very important third imperative in these types of arrangements.
Thank you so much. I think I could go on forever, and I'm sure a lot of our audience members could with questions. This project is so fascinating. But we are right on 10 o'clock in Australia and on the hour for all of our colleagues on the other side of the world. So I just want to join everybody and giving another thank you so much to Claudia, Jean-Michel, and George for their time today. That was phenomenal. I can also second how exciting and wonderful their earlier paper with Jack Kloppenberg was, and we're all very excited to see all the work that comes out of this project. So thank you so much for giving us your time today. And thank you everybody for joining and being a wonderful audience today.
Thank you so much for
Yeah.
the kind invitation, and have a great rest of your day.